Organizers for inviting me to come to this beautiful city, Bajaja, and share some of the work we've done over the years, specifically on malaria vector control. So on this first slide, I show my collaborators and also my students that have contributed to some of the work I will present here today. Their names are given in bold. Okay. This is a picture of our university. It shows a view to the western side. The building there, the tall building on the right-hand side top, that is the engineering building 1. Next to it is engineering building 2, and our laboratories were in the engineering building 2 on the left of it. Okay, so basically malaria research at our institute started around 2011 with a project that was funded by SASOL. And over the time, we have graduated 6 PhDs and 4 masters on this research activity. And we focused on controlled release of insecticides, larvicides, and mosquito repellents from polymer matrices. Okay, so in 2018, I already gave quite a comprehensive talk on the sort of work we did on malaria vector control. Our focus was very applied because we were basically developing products to fight malaria. And I'm listing here the products that we've developed for solid insecticides. Those were knitted polypropylene bed nets, polyethylene wall linings that are shown on the left, and the knitted polypropylene bed nets shown on the far right, and the fibers are showing there in the middle. And we also had a project funded by the Bill and Melinda Gates Foundation on a gypsum-based whitewash. Okay, so when it comes to liquids, additives, actives, we have developed microporous strands, bicomponent fibers. Those are shown there. That's a bicomponent fiber. It's got an EVA core and a polyethylene sheath. So this gives you zero order release because the outside sheath polymer acts like your membrane, but you trap a large amount of liquid additive inside a liquid imbibing polymer like EVA. We've also done tri-layer films, where the active is in here, and the outer layers then control the rate of release. We've developed isotropic repellent blends. Here's a picture of the lady who developed it. She's actually from Iran. And we've done some work on making in situ microcapsules to also trap actives inside and release them very slowly over a long period of time. Okay, so today's talk, I cannot repeat everything I did in 2018, so I'm only going to focus on 2 new areas that I've not talked about before, at least at this conference. And basically, it's about malaria and malaria vector control with vector control interventions as a target. And the 2 topics that I will address today are blooming of insecticides from polyethylene matrices and polypropylene matrices, and then also a little bit about green larviciding. Okay, so first of all, I must give you a perspective on what malaria is. It is a tropical disease. You can see here in the red areas here on the tropical regions, that's where malaria is a big problem. Clearly, you are very lucky in Algeria. There's almost no problem with malaria whatsoever. But in sub-Saharan Africa, it's a major problem. According to the World Health Organization, we have about 200 million cases per year and about 400,000 deaths or more per year. And the problem is that most of those that are dying are children that are 5 years or younger, and also to some extent ladies that are pregnant. So according to the WHO, the best strategy to control malaria or reduce the amount of malaria that is present would be to eliminate the malaria vector, which are the mosquitoes. So it's the mosquitoes that are transmitting malaria from when they bite you from one person to the next. And if you look at here, there's a picture which shows you that little organism that gives you malaria. It comes via the mosquito into your body. It first goes to the liver. It's got various stages, life stages. It first develops in the liver, then in your bloodstream, and it actually attacks the red blood cells. It's a unique organism because it is, the hemoglobin, which is an iron-containing polymer that is normally toxic to other things, but this organism can actually eat your red blood cells, and that is what happens here. So because the red blood cells actually carry oxygen to your body, if your red blood cells are reduced, you actually die from asphyxiation. So that is basically the problem of malaria. So if the purpose that we want to address is to actually control the number of mosquitoes, then we must look at the mosquito life cycle. So first of all, the mosquito female lays eggs. Those eggs are hatched and become larvicides that live in water, stagnant water. They develop and eventually become pupae, and in the pupae you develop a new adult. So the two targets that you can consider from the WHO perspective is to actually kill the adult mosquitoes with adulticides, and secondly larvicides. So, but there's also possibilities in future to look at preventing eggs from hatching. Okay, so now we need to look at what actives can be used to assist you in vector control in terms of killing the mosquitoes. And obviously you can get different compounds. You can get insecticides, larvicides, repellents, attractants. These can be either solid liquids or vapors. They have different modes of action. Some are contact poisons. Some can act as repellents or attractants. When we talk about attractants, we combine that with an insecticide to kill it after it's been attracted. Unfortunately, these compounds also have a problem that they also degrade in the environment by hydrolysis, by thermal degradation, by UV degradation, also by biodegradation. So then the way you can actually apply these would be by different delivery modes. You can use emulsions or dispersions, fibers, textiles, polymer mesh films, mats and tapes, etc. And the key questions that we try to address is how can you prolong the activity so that you use very little, because these things are expensive. Sorry, this is getting very loud now. Let me put it a bit further out. Okay, so if you look at currently used strategies, the first one is obviously topical repellents. You can go to the chemist and you can buy a repellent. It's either lotion or a spray that you put on your skin. And the problem with that, it only gives you a small time for effectiveness, typically not more than, say, 2-3 hours. The WHO is pushing the long-lasting insecticide-treated nets. This is, for instance, a net that people sleep under. The problem is it only protects you when you are sleeping. Then the other one that is actually applied very extensively in South Africa, Mozambique, Namibia, and mostly the countries in Southern Africa, is what they call indoor residual spray. And what we do is we actually spray mostly DDT on the walls of the buildings. We normally are fairly high up on the walls of the dwelling walls. You ask yourself, why will this work? Well, what happens is the mosquito that gives you malaria, the Anopheles mosquitoes, they tend to bite you when you're sleeping, typically after midnight. And then when the female mosquito, she needs that blood meal to make her eggs mature. But now you can imagine if you eat a lot of meat, you feel tired. The mosquito also feels tired after she's drunk a lot of blood, and now she goes to rest. And typically what she does, she goes and sleeps at the top, near the top of the wall. And she tends to spend 2 to 3 days there before she moves on to lay her eggs and then find another person to bite. So therefore, if she starts resting on this wall, which has now been sprayed, she will pick up some insecticide and she'll die. That is the way it works. The problem is why we use DDT is that the normal or less, the more benign insecticides, they are not stable on these walls. You can imagine it's either a concrete wall or it's a mud wall, and that has catalytic effects causing these insecticides to degrade very rapidly. So therefore DDT is used because it can last a full season, and even after a season the DDT doesn't work anymore either. Okay, so the first project we did was actually to develop a net. We were able to come up with a net that passes all the WHO requirements. Basically, the specification is that you must be able to pass 25 wash cycles without losing effectiveness. And our system that we developed was polypropylene-based knitted net, which contains about 0.57 wet% beta-cyfluorine inside the polymer that you are using to make the fibers for the net. Okay, so if you look at what those fibers look like, here are the fibers in electron microscope. What you will notice if you structure those fibers, you will see that there's actually kind of a skin structure on the fiber surface. Here is a piece of the skin that's been folded back. Here's an enlarged picture of that, so you can now see the underside of that skin. And if you do a confocal fluorescence microscopy, that corresponds to that. The insecticide is fluorescent, so you can see that the insecticide is accumulated at the bottom, below this sheath that is covering the fibers. And that is why you can actually wash it, because the insecticide is not completely on the surface. It migrates from the bottom up after each wash, and that way you retain effectiveness. Okay, so the way you test the bed nets is illustrated here. There's a specification from the WHO, which this is a cone bioassay test. What you do is you take a number of mosquitoes, you have a net material here at the bottom, you have this cone, you take the mosquitoes, put it in there, and then you measure the knockdown. Basically, knockdown means after a certain time, the mosquitoes fall over and they have their legs in the air. And after 24 hours, you check how many are actually dead. So the requirement is that you must have more than 80% mortality, and you must have 95% knockdown after 60 minutes. This is an interesting one. The WHO has discovered that in Africa, Southern Africa, Middle Africa, if a mosquito falls on the floor, within 1 hour, the ants will eat it. So knockdown is very important because then you actually use the ants also to kill the mosquito. So here is just a proof that we actually managed to get 25 washes and still pass the test. And so this proves that the system did actually work. So as I said before, we are using in South Africa no bed nets. We're only using this DDT spray. There's some details on why it is effective. You can actually get the WHO-approved insecticides other than DDT. You can actually absorb them on phosphogypsum, and you can make a whitewash, or you can also integrate them into clays, and then you can also get them to last longer. The problem is that you need a lot of phosphogypsum, you need a lot of clays. The clays become very expensive. This one is not expensive, but you need about 60 kilograms to treat one house. That is too much because in these rural areas, there's no real good access to dwellings because they're everywhere in the bush. So it's not a very effective way. So the question is, can we come up with a better idea? Can we maybe combine the spray in some way where you actually have something on the wall with something which is a bed net, but it's not covering the people? And the idea then that we came up with was to actually use a durable wall lining. So the durable wall line, here we show a picture of one of my students installing it into a house. We did a field trial with 40 houses where we installed this. This is what it looks like after it's installed. So here's a mesh that is put on the wall fairly high up because you only need it fairly high up as the mosquito likes to go to the top. And so how does it work? Well, here are some details on why it's very effective. Compared to indoor residual spray, our field trial ran for 7 years. And after 7 years, we still had 100% effectiveness of this. It was still killing mosquitoes, even though the amount of insecticide was very, very low that was incorporated into the plastic mesh. So the reason is that the polymer they're using is basically keeping the insecticide stable. It doesn't degrade, and therefore it can last a very long time. Okay, so here is the process whereby you make this product. It's the cheapest way of making a mesh or a net. And effectively what you have, you've got an extruder, and these are typically made from polyethylenes. And in this extruder die here is actually 2 concentric cylinders that turn in opposite directions, but there are grooves machined at an angle inside to these on the outside of the inner cylinder and on the inside of the outer cylinder. And as they turn, the polymer flows through the grooves. And when they come to the outside and you cross over, then you touch the 2 strands and you make effectively a net. Then you pull it down over a shaper here, and then you can also stretch it afterwards. And in the end, then you end up with a net that is some width. You can make it up to about 1 meter wide. Okay, so what is the trick here? The trick that happens is that you must have an insecticide. So it doesn't work for all insecticide. You need an insecticide that is virtually insoluble in the polymer matrix at room temperature, but highly soluble in the melt. And then what must happen when the polymer cools down, the insecticide must bloom to the surface, and it must form crystallites that stick to the surface. And these crystallites, if you want to wash, you must make sure that you end up with a crystalline solid on the surface, otherwise you will just wash it off. Now, as I said, with our Netlon-based wall lining, the mesh that we make is a Netlon-based process, and it can remain effective for at least 7 years. We stopped the field trial after 7 years. We don't know how long it really will last. Okay, so just to show, if you use different insecticides, we did a very large range of insecticides. Here I'm only showing 3, alpha-cypermethrin, fipronil, and chlorphenapir. And, alpha-cypermethrin is highly effective. You need very, very low concentrations. These 2, you need higher concentrations. But here is fluorescence microscopy, and you can see spots of the alpha-cypermethrin here. And here you can see with the fipronil and chlorphenapir, because there's much higher loading, the amount that has bloomed to the surface is much, much higher. Right, okay, so really then the trick here is that you must have a blooming of the insecticide, that it must accumulate at the surface, because this is a contact poison. The insecticide must touch the insecticide and pick up some of the molecules, and then it will die. Okay, so here's schematically the blooming process for an amorphous additive. The blooming is sometimes a problem with other, for example, flame retardants can bloom, and you don't want that because then you get a discoloration on the surface of your plastic part. But in this case, we employ the blooming, but for an amorphous material that is blooming, amorphous additive, initially there will be homogeneous distribution of the additive inside the fiber, for example, and then as it cools over time, you actually get that it migrates to the surface, and then you get sort of a coating on the surface. So this is the idealized representation how that works, okay? And in reality, it looks a little bit different. So we did quite extensive study to analyze this, what happens for different insecticides. Today, I will just give you some information on using polymer LDPE, low-density polyethylene, high-density polyethylene in a 1-to-1 blend. And here I will also talk about only 3 insecticides, alpha-cypermethrin, chlorphenapir, and fipronil. And these are the ways we characterized the material that we generated. So as you saw before, you can see that the stuff has actually bloomed to the mesh surface. But the problem is if you want to characterize that, it's very difficult to deal with fibers which are very thin, 20 micron or so. And for mesh, it's very rough surface. So what we decided instead for characterizing the blooming is to actually blow film. So incorporate the insecticide in a blown film. And then what we did is we used FTR ATR. So this is schematically an FTR ATR system. You have the light coming in at an angle, total internal reflection. This is the green is your sample. And you actually pick up with the ATR only the material which is right at the surface. It's kind of a quantum effect that the light, even though it's total attenuated reflection, you get it attenuated because of a quantum effect. It actually sort of goes a little bit into this and then comes out again. So how did we do this? Okay, first of all, let's just look at the 2 spectra. This is an example for chlorphenapir. This is the spectrum for chlorphenapir pure powder. This is for the polyethylene pure film. And you can see there are bands, IR bands, which are different. So you can use, remember when we actually add the insecticide, we use very low concentrations, less than 5%. But here is for the pure components. So you can use this as a reference to get an internal reference concentration of the insecticide in the polymer, because you reference it relative to the polymer peak that you got here. But then when you look at an example of a film that was aged, this is the spectrum of the neat chlorphenapir, and then over time you can see how it grows after some aging at 40 degrees. So you can follow how much of the material is actually moving to the surface by FTIR ATR. Okay, so now it was important to determine how much of the insecticide stays inside the film, because you must obviously add more, because if it only stays inside and not come out, it will not be effective. So the way we did that is we made films with different concentrations of insecticide, and then what we did is we aged it at 40 degrees C in a convection oven, and we then did FTIR in transmission mode. So you first take the film after some aging time, you take an FTIR, then what you do is you take acetone and a smooth paper or cloth and you wipe it off with acetone. Then you can do an ATR to make sure you've removed everything on the surface, and then you do another transmission. So the difference in the intensity of those signals can then be used to estimate how much of the material was still inside and how much was outside. That gives you then graphs like this, where you can see how, depending on the aging time, how does the amount change. You can see there's a plateau for the amount remaining, which tells you that this is kind of an apparent concentration of material staying inside the polymer matrix. So the minimum dosage you must have would obviously then be about 0.2%, otherwise it will not come to the surface. And so this was the way in which we determined what is the amount of insecticides that remains behind and how much comes to the surface. Now I must say that it's not a solubility issue, because some of the insecticide will actually crystallize inside the polymer film. So for example, that's why we use HDP, which is highly crystalline, up to 90%, and LDP, which is much lower crystallinity, like 40%. And you notice that in the HDP, a much higher percentage remains behind. It's obviously getting trapped because of the fact that the crystallites do not allow the insecticide to diffuse through the system. Okay, so just to look at what it looks like if you leave those films for a very, very long time. Here is a picture of alpha-cypermethrin in HDP, alpha-cypermethrin in LDP. It's not exactly the same picture. And here within LDP, you've got torfenapir and fipronil. So you can definitely see with the optical microscope that you do get surfaces. And as you can say, that blooming picture I showed before shows like a homogeneous deposit. That's not true. You form sort of dendritic crystals in the case of the alpha-cypermethrin developing on the surface. And they're not easy to wash off. They stay there. You need to have an organic solvent to get them off. Okay. So we also did some SEM-EDX to do elemental mapping to show that the stuff is actually at the surface. So the green color that you see here at the bottom, that is chlorine, using chlorine to identify where the insecticide sits. And these were two different films with different active ingredients. Okay, so in overall conclusion from the blooming studies, what we found, and obviously I'm only presenting some of the characterization techniques because of limitations in time, but what we find that the blooming process levels out after about 4 days of oven aging at 40 degrees C. Independent of what insecticide you use, you find that HDP traps a lot more than LDPE. Optical and SEM imaging of the film show that there are insecticides present in discontinuous flake-shaped and dendritic formations on the surface after a long time. And the insecticides do not affect the crystallization process of HDP that we discovered by doing DSC studies. And what we found is that you can use as little as 0.29% of alpha-cypermethrin and you will pass the WHO test that is also applied for bed nets using the meshes. Okay, so there's some detail here, but I think I'm running out of time, so I'm going to skip that bit because I want to really just talk a little bit about trying to do larviciding. What we've just found now is that the number of malaria cases have stabilized, they don't increase anymore, and WHO has identified that we need to find additional mechanisms of getting vector control going. And one of the things that they're now pushing is to go for larviciding. So the mosquitoes breed in stagnant water. They don't breed in rivers that flow, only in portions and lakes where there's quiet water near the wall, near the banks. And when you have the eggs deposit in the water, within as little as 5 days, but usually a bit longer, you actually generate a new generation of adults. So unfortunately, there's a lot of water around. I mean, even tire water lying in tires that have been discarded in the field, that little bit of rainwater that goes inside that tire is sufficient to grow mosquitoes. So what we found then is, my students, when they did study to see what would be a useful and less expensive larvicide, everybody was, if you look at the literature, there are hundreds and thousands of papers on using essential oils as larviciding, and they're very effective. They work at around 10 ppm. But the problem is, they come from plants, they have to be extracted, they're very expensive, doesn't make sense. And what we discovered that it's actually possible to use oleic acid, which is a food-grade constituent, and it's also derived from plants, and it's soluble in ethanol, and that's it's known to be an insecticide or larvicide for some mosquitoes. Now, in our country, the Anopheles arabiensis is actually the one that gives you the malaria. There was no data on Anopheles arabiensis, so we did the standard test of the WHO to determine what is the toxicity of this one, whether it is actually effective larvicide. If you look at these graphs here, you can see we dosed different oleic acid dosages in water, and we then measured by a standard WHO bioassay test, what is the mortality. You can see with biosystems, you typically have this, that you've got a huge variability depending on how you do experiments. But still, if you do a proper statistical analysis, and this is obviously a huge amount of data that's summarized here, we found that the LC50 is about 13 plus minus 21 ppm. Okay, so how do you now get oleic acid? You can't just put oleic acid in water, it will just make a thin film on the surface. So you need to have a way, and if it's exposed to the surface, obviously it will UV degrade because there's a double bond in oleic acid. So you want to find a way to actually get the oleic acid in water dispersed, but slow release of the oleic acid. And so the system, one of the systems we developed is to make use of Zane, which is a polymer, This is corn or maize, and from that you can extract the protein, which is pure Zane. It looks like that. And if you can actually dissolve it in mixtures of ethanol and water at a certain concentration, you get it to be soluble, and if you let it evaporate, you make a nice, beautiful Zane film, and if you plasticize it, you also get very nice mechanical properties even. So it's a very unique protein because it is typically non-polar, but it has groups that can be protonated, and it's also got carboxylic acid groups, and it means that it has sort of like surfactant-like properties. So one part of the molecule, when it's folded, is oleophilic, and the other part is lipophilic, and it makes it then possible to play all sorts of games with it. And here is just this illustration. There's been a lot of fundamental work being done. If you look at the folded form of the Zane, they write it down like something like this, and portions of it will be hydrophilic, portions will be lipophilic, and so you can actually then either stabilize hydrophobic domains either by varying the pH or by varying the solvent and non-solvents that you use when you do the trick. But you can, with the same polymer, you can also stabilize hydrophilic domains. In other words, the Zane will absorb on hydrophilic or on hydrophobic, depending on what conditions you use. So there's a huge amount of information locked up here. I'm not going to go into details, but we decided to use this system then to see whether we can make a fully biodegradable, from renewable resources based larvicide. So the idea then was to use, if you go, maybe I should just go back. You can see here, one of the possibilities that you can do is to make Pickering emulsions. That relies on making the Zane particles becoming nanoparticles, and then they absorb on the surface of your droplets. That would obviously be this would be the oil. And so just conceptually, what you want for a Pickering emulsion, you don't use a surfactant. What you actually use is you're using particles that have got Janus-like properties. So one side of the particle likes to be with an oil phase, the other side likes to be in water phase. So if you've got an oil droplet, like here, say a lake acid droplet, then you can cover it with these particles, and that will give you then a stable emulsion droplet. Okay, so if you then look at the phase diagrams, the Zane is soluble in mixtures of, in this region, the Zane is soluble, and in this region, the ethanol is water mixtures, lake acid. So lake acid fully soluble in ethanol is not soluble in the water at all, but as you as you add water to the ethanol, you can go up to here, for example, at that level of water, it will still be soluble. But looking at these two phase diagrams and doing some interpretations, you can come up with a formulation that will give you a Pickering emulsion. And this is just a summary of the system that we have developed and ended up with. And the trick was then to make a 10% Zane stock solution and 80% ethanol water. And then you mix that with 30 parts of Zane stock, 30 parts of that with 30 parts of lake acid. And then you stir that up, and it actually makes a homogeneous solution. Then you high shear mix that in 40 parts water. That ends up giving you a Pickering emulsion, which has 30% of lake acid present. And the Zane particles are in the nano size, and they are formed in situ during the mixing process. And they cover the droplets, and the stability of these emulsions are several weeks. So they're not permanently stable. They also break down over time, but you can get several weeks of stability. And then you can take this emulsion and dilute it by a shear mixing process into larger quantities of water. So this just shows you a picture of the lake acid droplets. You can't see the Zane particles here, okay? But this contains 3% Zane in the end and 30% of lake acid. And we also then did bioassays on here that is required by the WHO, the World Health Organization. I'm not going to read through the details here. I can give you some papers if you want to read it. But basically what you do is you dilute it to certain concentrations. And so we did tests where we did a glass container that we filled with 100 milliliter of a diluted emulsion. And then what you do is you add 25 mosquitoes larvae to that. And then you measure the mortality after 24 hours and 48 hours. So this test has to repeat it on 3 different days, and each day you have to do 4 replicates to actually get good statistical values. That is a specified protocol that the WHO has come up with. Okay, so we actually did one additional test, because initially when you do these bioassays, you have to make use of facilities elsewhere, and you want to be sure that you actually have some results that are useful. So we also cheated a little bit, because we actually also just did the standard Pickering emulsion, and we did another emulsion where we added a small amount of sodium oleate, which is a co-surfactant, which apparently is supposed to assist the Pickering emulsion to be even more stable. But these are the results then for the 24-hour mortality at the doping of 250 ppm malic acid in water. You get, as you can see, 98%. That's way above what you require to be effective. And yeah, so in conclusion, what I can say is that blooming is a very important principle which is responsible for the activity of contact insecticides when they are incorporated in textile fibers or in plastic mesh. And I also would say that it might be very well be possible to formulate very inexpensive yet highly effective mosquito larvicides just by using benign constituents. But our work has been limited to laboratory studies. We still need to do a field trial to make sure that that will work in general. So I just want to also show some pictures of the students that were involved in this work. Okay, and here's the group of people that installed the mesh in the far north of our country. And, you know, we did a one-week trip to do that. And then I have to acknowledge the people that sponsored our research. These are listed here. And then I'm also ready to answer some questions if you have any.